The overall goal of this procedure is to shorten the classical Yamanaka IPS approach by directly generating induced neural progenitor cells from a patient's skin biopsy for therapeutic applications. This is accomplished by first performing a punch biopsy from the patient to derive primary fibroblast cells that can be cultured in vitro. The second step is to expand and subsequently infect cultured fibroblasts with viruses that encode reprogramming factors to induce transdifferentiation. Next, the putative converted induced neural progenitor cell, or INPC colonies, must be carefully selected by visual validation and subsequently isolated by manual picking approximately 20 days after infection. The final step is to monoclonally expand the INPCs in neuroinduction medium. Ultimately, targeted in vitro differentiation, as well as immunofluorescence microscopy, is used to show that patients' own directly converted INPCs are able to differentiate into neuronal as well as glial cells, making them a virtually unlimited source for biomedical applications.
The advantage of this direct conversion technique over existing methods, like the Yamanaka-type derivation of induced pluripotent stem cells and their subsequent differentiation, is twofold. Direct conversion into multipotent induced neural stem cells, we call them INS cells, is not only considerably faster, but also safer. Thus, the transdifferentiation procedure will make it much more achievable to produce patient-specific cells for therapeutic application.